Okay, so what I'm going to try to do is not so much try to tell you what genomics is about. I know many of you already know, and I don't want to presuppose that I know more about genomics than anybody else here. I do a lot of presentations of genomics for local high school workshops and also at the Franklin Museum where they had the STEM Scholars program. So this is sort of my 10-minute spiel that I give to the high school students when I try to do a genomics lecture. And just as a point of reference, these lecture slides I have now put into the shared folders so that if you want to use any of these slides in your own classroom, please feel free to do so. So the first thing I start out with this age-old question of chicken or the egg. And the question is, what comes first, chicken or the egg? And really, from the genomics point of view, what really comes first is the information that makes the egg into a chicken. So it's really that, and that information actually is, of course, in the egg, which contains the genome, which is made of the DNA. And this is a very large molecule, which somehow, through lots of chemistry and physics, tells this egg to become this complicated organism called the chicken. So maybe in some sense, the answer is that the egg does come first. Okay? And so that, of course, we all know the DNA molecule. And what's actually remarkable about the DNA molecule, which this picture comes out in many textbooks as well as covers of textbooks, and this is something that I teach to my own college students, is that this molecule is one of the few molecules that we know in nature where the shape of the molecule does not change dependent on the atomic constituents of the molecule. Okay? What I mean by that is, as you know, that there are individual molecules here which we call T for thymine and adenine and cytosine and so on. Okay? And we know along the DNA string, all kinds of different, you can have T, 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 A, G, G, all kinds of orders of these molecules happen. Normally in chemistry, when that happens, then the whole shape changes. Right? So if you change the chemical composition of the molecule's shape and physical properties change. DNA is one of the few molecules we know where this does not happen. Okay? And what that means is that our shape is invariant to the constituent parts, and so what really comes out of this is not the chemistry or the physics, but actually the information. Right? This is a really remarkable fact about nature, that what we are is encoded information, and what happens in the computer is not too different from what actually happens when a cell takes all the information and creates a chicken out of the egg. And people have done experiments on these DNA molecules. If you change some of the physical characteristics, then it suddenly becomes, the shape changes with all the constituent parts, right? So nature has evolved this molecule to carry information and own the information, and this is really one of the most remarkable facts about DNA and the genome. Okay. So genome, of course, is a sequence of letters that we think, which are actually molecules that are strung together, what we know as nucleotides. And like the English letters, the particular sequence matters. So BAT is not equal to TAB, and we know that ACT is not equal to, say, TCA in terms of what it does in the cell. And so therefore, we want to understand this order. The human genome is comprised of 3 billion DNA letters. Each genome, if you fully stretch it out, is about 6 feet. And so if you take all the cells in your body, there's about 100 trillion cells. You take all the DNA that each cell contains and lay them out end to end. It'll reach the distance between Earth and Moon 50 times. So this is the amount of information that's in your entire body. Right? By the way, I think one of you said that you wanted to, this connection between this white stuff that you do in the experiment and the information. I have some slides at the end which you can also use. One of the things I try to do a lot when I do the high school sessions is to try to talk about the scales and how difficult it is to go from that white stuff that you see to the actual molecular parts like A, C, and G, and what the difference in the scale will be. And we'll go over some of that again when I do the DNA sequencing session. Yes, Larry?
You know, I've seen this a million times, but now I don't know, for some reason I'm coming up with a question. So just to be clear, when you say each genome is 60, are you talking about all, like one set of 46 chromosomes, or are you talking about one DNA molecule of one chromosome?
We're talking about all 46 chromosomes and late
From
length
one cell.
Yeah, so if you take the DNA, you know, as you know, DNA is all wrapped up in a tiny little core. If you stretch it out and then stretch out those 23 pieces, then that's about 6 feet. So the human body has about 100 trillion cells. So nearly every cell has a copy of the genome, 3 billion letters. So there's this many letters of DNA in your body. This is 10 to the 23rd. So this is supposed to be 370 million. And you have to Google that word to know what that is. And apparently, there's about 370 million stars in the visible universe. So you could say that our body has as much information as there is stars in the universe. Okay. So these 3 billion letters in the genome has information about how the cells work and how to make the tissues out of the cell and how those tissues should work to make organs and how the organs make us. Okay. So it's like an instruction book for our bodies. Okay. But the main problem is, and this is really what we do all the time in our research genomics, is that this book is written in language for which we only know a very small number of words. We don't know all the words that are in this book. We know a little bit of punctuation. Okay? That means that we don't know where the information starts and ends. There's 3 billion letters that are in a row, right? Some, you know, we know that the information comes in little packets. We don't know where that punctuation is. And we know some sentences here and there, right? So the genomics is all about learning how to read this book. Now, because of this, we want to first physically read the book. And so this is DNA sequencing. I started in college in the early 1980s. And I actually worked at sequencing actually from this fungi organism. And at that time, you had to work about a year to get 600 letters of information out. So that's about 1.5 letters per day, right? Now, Genomic Technologies, in the last 40-some years since I started college, everything sort of evolved to be faster, smaller, more precise, less expensive. And so, you know, I used to do experiments with this kind of test tube. Then we switched to these kind of things, which are the microtube. And then these are 96 well placed. So each and every time we make the chemistry smaller and do it multiple in parallel at a time. And these became even smaller so that people started using technologies that are the same technologies that we use to make computer chips, to make little chemical reaction chambers. And eventually, we made them so small that you can't really see them at all, and you need a microscope to look at it, and you're able to run chemical reaction, billions of them at the same time. So not just 1,000 or million, billions of chemical reactions at the same time. And so now with this next generation sequencing, we are able to sequence 300 billion letters per day. Right? That's kind of about 2 letters per day to 300 billion letters per day for each one of these machines. And we have many such machines. Right? So when the Human Genome Project started, it cost several $1 billion to complete the project. Now we can do it for under $1,000. When the Human Genome Project started, and when people talked about sequencing, say, using the technologies way back then, it didn't have, if technologies didn't advance with the project, it would have taken about 3,000 years to complete the project. So you can think about what those people were thinking about at that time, saying, let's do this. Right? It'll just take us 3,000 years, no big deal. So this is just remarkable that people are able to push forward such a project as well that the technology developed along with it to be able to do it. So new data is currently being generated at something like 4 trillion letters per day. We know that sequencing has now become going into the clinical practice. Once that becomes routine, we'll probably have 1 billion genome sequenced, not too distant in the future. And even with 1 million full-time scientists doing nothing but looking at the DNA, we know that less than percent of the data will actually be seen by human eye. So what we do is, in fact, use computers and modeling. And this is actually on the research side what I do most of the time. I do computational biology and modeling. So the take-home message is that, in fact, computation and mathematical analysis is currently, and will be even more so, essential to modern biology and also practice of medicine. So what biomedical scientists used to do is start with some disease or symptom, like say autism, and figure out what organs or systems are relevant to that disease, and then figure out what subsystems underlie that disease, and then eventually try to figure out what molecules or genes are responsible for, say, autism and so on. But the genomics research program is to invert this process. We start out with enumerating all the genes or the molecule, because we're able to use this economy of scale and be able to do simultaneously with so much cheaper. Then we try to quantify those molecules, enumerate all the interactions that the molecules are doing. Then we model the system function of those parts, what they're doing as a system, and eventually try to get what they mean for the function of the organism or the dysfunction of the organism. So, for example, we now know 2 brain tumors that look the same. They respond very differently to treatment. Things that we used to think to be the same tumor, now we know comes in like 5 or 6 different molecular flavors, each of which respond differently to different kinds of, say, chemotherapy treatment. So genomics and the idea of precision medicine is really revolutionizing how people see diseases and how we approach their treatment. So genomic information allows prediction of disease risk, understanding the molecular basis of disease, and also designing new drugs, of course, as we understand more about what molecules are affected by diseases, and prediction of individual responses to disease drugs, designing new biology with engineering precision. This is that field of synthetic biology that has just come into being in the last 10 years or so, where we can try to use engineering principles and then use technologies like CRISPR to try to design things like some functional outcomes of organisms. So genomics is this new way of understanding biology and diseases. The key word here is by systematically studying what we call the information part of the genome. Right? So systematically meaning that we start with first enumerating all the parts there are in the system, and then try to see how these things all work together in the system. All right, thank you very much. That was my short introduction to genomics. And as I said, I have many slides in here, especially things that emphasize scales of things and how difficult it is to work with scale. For example, if DNA molecules were about the size of a grain of rice, then your hand is about the size of the continental US, right? And I do that because your hands are what you use to manipulate things, right? Grain of rice is about what you can pick up with your hand. And imagine trying to pick up a single nucleotide, right? This is like having a hand the size of the continental US and trying to pick up a single grain of rice, right? And this is why, of course, molecular biology is really difficult. It's really all about dealing with very, very small things. Okay, thank you again.